All right, so last time when we left off, we were just getting started talking about the inflammatory pathways. So I'm going to pick up with phagocytosis here. And I know that we've talked previously about, and we got into that already, phagocytosis and kind of generally what it is. But I want to touch a little bit in a little bit more detail on the steps in phagocytosis, because learning about the importance of phagocytosis in our innate response helps us kind of understand how we're going to lead into our adaptive response next.
This is 15 still, right?
Yes, this is still 15, yes. So phagocytosis, phago eating, cytosis, cells are doing the eating, right? We have the steps in phagocytosis. The first one is chemotaxis. And we've mentioned this concept previously when we were learning about this in Unit 1. But chemotaxis is chemochemical taxis movement. We're moving in response to chemical signals in the body. So when you injure yourself, let's say you stubbed your toe and you got a little, ouchie boo-boo on your toe and you're bleeding a little bit. Now your cells in that area immediately start sending out signals for help. Those signals for help are going to draw in our inflammatory cells and the molecules that come with them that are involved in the healing process. This whole process of being drawn in by that chemical signal is chemotaxis. Alrighty. Then once we've drawn in the cell, we're going to have our white blood cells start ingesting things in the area that are non-self, or damage from, like when you start rupturing open cells from stubbing your toe. You've got damaged cells. Our white blood cells will come in and eat up all the debris, bacteria in the area, the dirt in the area, and things like that, and just ingest it. What happens when we ingest as a phagocyte is we create a little vesicle called a phagosome. So when we think about zome, that's just a way of saying vesicle. So a phagosome is the eating vesicle. The first vesicle you get put in, you get eaten up through phagocytosis. That phagosome is going to be enclosed and encased inside of the cell. And we've talked previously about another vesicle called a lysosome when we were learning about our cell parts. Our white blood cells, they have lysosomes that have things like lysozyme in them, but also acid and reactive oxygen molecules and things like that. All of these are chemical defenses inside that lysosome that will kill and break down whatever we ingested in the phagosome. When we merge the lysosome, the toxic compounds in it, with the phagosome, or we had the thing that we ate up, we'll merge those two together, and that creates the phagolysosome. So it's just merging those two together. Then those chemicals will act on the organism that we ingested, bacteria or the debris or whatever it was we ate up. And usually we can kill bacteria within about 30 minutes in this process. That's the destruction step, and we'll usually excrete out that waste that we broke down. This is pretty much what neutrophils are going to do. Neutrophils are main responders, the most populous of all the white blood cells in your blood. The neutrophils are going to be your first responders as phagocytes to the site of injury or infection. They're going to show up and just eat anything in the area. They're going to eat all the bacteria, all the debris, any damage from the cells, anything that's not an active, living, healthy cell, they'll just start ingesting it. And they'll just be excreting out all of their waste. So they're just pooping out everything that they just broke down and leaving it in the area. They do this pretty generally. It's pretty nondescriptly. If you're not a healthy cell and you can't confirm that you're a healthy cell, then we're just going to eat and break it down. All right, and I can recognize that through, yes, questions, Shy. Phagolysosome? I think I have a slide on it, so let me get to that in a second. So being able to recognize first that the pathogens are not self, we can recognize them through those PAMPs, as well as we have markers on our cells that we can be trained to identify as self markers. So that's going to help with identifying things and helping know what we should ingest and what we shouldn't. The phagolysosome. So the phagosome is a vesicle that we are ingesting the thing in. The lysosome is a vesicle that contains those toxic chemicals, and they merge together to create a phagolysosome. So once we have the eating vesicle merging with the toxin vesicle inside of the white blood cell, once those things merge together, it's like whenever you're finally meeting, like you eat food, think of your mouth as being a phagosome and your stomach is the lysosome. And then once we get that food mixed in with what's in the stomach, that's your phagolysosome doing all the work on what you've eaten. Does that make sense? All righty. So that will lead to death within usually 30 minutes. And that's the gist of phagocytosis. And we're just going to go around eating non-self, debris, and pathogens, anything that can identify itself as a healthy self cell, and then excreting out the waste. We mentioned our signs of inflammation: rubor, calor, tumor, and dolor. That rubor is the redness. Like if you speak Spanish, these are probably easy terms for you. But if you don't, the Latin rubor for redness. This is caused by the blood flow increasing into the area after the injury. So it turns red because blood is red, and we're increasing the blood flow to that area. The whole reason we're increasing the blood flow to that area is to move our cells that are transported by our bloodstream to the injured area. And then they will move out of the bloodstream and into the tissue with all of the fluid and the nutrients and other chemical signals and things, that chemicals that are involved in healing, all of this stuff moving out of the bloodstream and into that tissue. That movement of the fluid out of the bloodstream and into the tissue causes swelling. That's the tumor. Increased blood and all of this warm stuff that's coming out from straight from the center of your body out to your extremities after you've injured yourself leads to warmth. We also have an increase in temperature, which we're going to talk about with fever here in a moment. That serves a purpose to increase the metabolism of our white blood cells and to help impede microbial growth as well. And then the dolor is the pain. When we have stimulated our nerves, the reason that we need to have a pain response in our extremities, especially, is just letting your body know that there is an injury, that there is inflammation, that something is going on that you might want to be aware of. And we see, effects of that in people who can't feel pain, being at severe risk of having injuries that they don't know that they have and can't care for properly. A lot of times with an inflammatory response, we can also have some loss of function. If you injure your back and then you can't move for a little while, that inflammatory response has kind of inhibited a functionality of maybe your back muscles and things like that. That can be associated with that inflammatory response. The whole point of inflammation is to bring our neutrophils into that area and all of our healing molecules into that area. But if you have too much inflammation, that can be a problem too, because neutrophils are going around destroying whatever they can, because a lot of these molecules can be involved in getting rid of potentially injured cells or damaged cells as well. You could also have some collateral damage involved in the inflammatory response. And this is fine and something that we can manage. When you stub your toe, if you have some collateral damage of healthy tissue in the area, it's not a big deal for most of us who are otherwise healthy. If you have long-term chronic inflammation, then the collateral damage builds up over time. Additionally, because we have all these signaling molecules that are involved in telling your body to start fighting the good fight constantly, other cells are coming in and getting signals, yeah, we need to be fighting, when they may not necessarily need to be. And this can lead to autoimmune disease. Getting the signals of we're supposed to fight when they're not supposed to be can also lead to signaling your cells to be fighting against self cells. We'll talk a little bit more in detail on autoimmune diseases in chapter 17 when we get to that one. Cool. And this can lead to damage of tissue over time, and it can be permanent as well. The whole point of inflammation, not all bad. It can have bad aspects if it goes on for too long outside of what its purpose is. The purpose is to mobilize the immune response to the area. Start getting our innate cells, like our neutrophils there, just start eating up and cleaning up the area very nondescriptly and start fighting with something. Because when we start needing our T cells and our B cells, our bombers and our tactical support cells, those guys are going to react very specifically to a specific antigen, but it takes a long time to mobilize and get those guys in the numbers where they're going to be effective. So that takes a long time. That can take a week, 2 weeks in order to get an active full response to something. We don't have time to wait on that. And your innate response is there to kick in immediately and get rid of what they can in the meantime to prevent infection from spreading and continuing until your T cells and B cells can get activated and get there and start fighting. Excuse me. All right, so the major steps in inflammation. First things first, you've stepped on a nail, apparently. And this nail brings in with it debris from the environment as well as bacteria. Well, in those tissues, all throughout all the tissues in your body, especially at the surfaces of your body, we have mast cells. We've talked about those being granulocytes, right? So mast cells are just hanging out there. They don't have a ton of ability to fight off an infection by themselves. However, they will release chemicals that will call in our big fighters. So they're going to release things like histamines, our cytokines, our chemical signal molecules, to call in for more help. These guys are like sentries. They're just hanging out looking for invaders. If they see an invader, they know better. They know that they can only fight to a very limited extent. So they're immediately going to call for backup. All right, so we've called for backup. Our bacteria are starting to invade into our system, and our backup shows up. What's our backup come in the form of? Neutrophils, primarily. So we've called for help. Those signal molecules have brought in neutrophils through chemotaxis, like we mentioned previously. And the neutrophils just come in and start eating up bacteria and debris. They also move in with plasma. The fluid from our blood will move into that area as the cells are squeezing their way into the tissue. And the fluid comes with things that are going to help mediate the inflammatory response, as well as molecules that are involved in healing, creating clots to stop bleeding, and all of these things. We have mediation of blood flow to and from the area as well with these chemicals that are being secreted by all of these cells involved in the immune response. Whether that is vasoconstriction, we often have decrease of blood flow to lead to like lessening blood loss, as well as to prevent microbes from being able to travel as quickly through the blood if there was injury in your capillaries and things like that. But then vasodilation, once we get the appropriate signals, to increase blood flow to the area to bring in our white cells. And that's all going to be mediated by those chemical signals. Finally, we've had all this fluid move in, we've had all of our neutrophils move in, and we are eating up all the bad guys. Now we're formulating pus, and pus is primarily made up of neutrophils. And there's some bacteria in there as well as other debris from the injury site, but it's primarily going to be neutrophils, as well as the fluid, the exudate that's moved out of your bloodstream in the form of the plasma and all the molecules involved in the inflammatory response. We have our clotting factors leading to the formation of a scab, and our neutrophils are going about eating up all the bacteria and the debris, the dead cells, things in the area, and basically excreting anything from that. And once healing has continued, we can form scar tissue and multiply our cells in that area to replace where the scab had formed. Scabs are meant to be temporary. They're just a way to block off bleeding and loss of fluid, but they will eventually be replaced by cells multiplying and replacing them. Your body doesn't care that you look pretty afterwards. Your body just wants to keep invaders from coming back in. And so it's doing its best to regrow that tissue to replace what's happened from the wound. Now, at this point, in this later aspect of our inflammatory response, we've brought in macrophages. Remember, monocytes that are in the bloodstream will move into the tissue and become macrophages or dendritic cells. And these guys are other kinds of phagocytes. They will go in and start eating up all of the debris that the neutrophils left behind, any cells that have lost their lives in this process of this fight. And the macrophages are going to go around and eat those guys. And the macrophages are essential and aiding in the next step of the process, that acquired immune response that we're going to talk about for chapter 16. So in chapter 16, when we start talking about our B and T cell response, your B and T cells don't just get activated because you have inflammation. They have to be activated by a specific antigen. Each B cell and each T cell that you have is specifically programmed to react to one specific antigen, not multiple. You don't have a B cell that recognizes all these different things. You have a B cell that this B cell recognizes this part of the COVID spike protein. You might have another B cell that recognizes a different part of the spike protein or a different part of COVID altogether, or a B cell that recognizes HIV, not COVID at all, and whatever. So every single B cell that we have in our body, each one from its original naive form, each one recognizes something different. And they're waiting out in your body right now, in your immune system, just waiting to see the guy that they were programmed to fight. And they're only going to respond to that guy, and only that guy can activate that. But they're waiting on that for all of that. And they're basically going to be waiting in your lymph nodes, often those B cells just sitting in there at our checkpoints in our lymph nodes, filtering out all that fluid that's coming back into the body, has to be filtered through the lymphatic system first. And they're waiting there in the lymphatic system, just looking for that one guy that they were made to fight. And when they see it, then there will be a series of activation steps that will allow them to drop their bombs, the antibodies. They won't do that until they get the go-ahead from the chief, which we're going to get into later. But those guys aren't ever going to see the one guy that they were programmed to look at unless they come into contact with it. And so that's either because the bad guy came in through the lymph and was being filtered out there directly, or because a macrophage at the site of exposure ate up parts of the bad guy and brought it to those cells. And then those cells can get activated because the macrophage brought it to them. The macrophages are really important for taking debris from the initial site of infection or exposure, wherever you have injury. They take the debris from there and go and check with all of those B and T cells and make sure, if you guys recognize this, I find this red marker cap hanging around in the tissues where there was an injury. Does anybody know what this is, and do we need to react to this? That's what macrophages are going to do for us. All right. Alrighty, so we've talked a little bit about what happens with the vasculature in response to our inflammatory reactions, whether we're increasing blood flow and decreasing blood flow. And this is all going to be mediated essentially by the chemicals that are produced by our white cells in the area, as well as potentially the injured cells themselves in those areas, sending out help signals. And that these things can lead to increased blood flow to the area. And when we have increased blood flow, the fluid can move out of the bloodstream and into the tissue. That's called exudate. Exudate is anything from the blood that's moving out from the vessels and into the extracellular space in the tissue area, not in the bloodstream, not inside of the cells of a tissue, but in the area surrounding the cells, the extracellular space. And that fluid doesn't normally belong there. And it's only moving in there as part of our inflammatory response, which is why we have swelling. It's not swelling. That's why we don't have normally the ability to contain that. You know, it swells up to accommodate that fluid. But all that fluid contains proteins, clotting factors, nutrients to help drive the inflammatory response. It can be clear, or it can contain erythrocytes, which is your red blood cells, or pus, where we have a high number of neutrophils building up with that, as well as potentially bacteria in the area and so forth. That swelling that results from that exudate coming into the extracellular spaces is called edema. And remember, the only way to get that fluid to get moving back into your body and get filtered out before it rejoins your bloodstream is through the lymphatic system. And the only way that that fluid moves through the lymphatic system is by skeletal muscle movement. All right, so here's diapedesis. This is our movement of our white blood cells out of the bloodstream and into those extracellular spaces in the tissue. And that this process is essentially squeezing between the cells in the capillaries where the walls are very thin, single cell thick maybe, and they literally just squeeze through the junctures between the cells. That's diapedesis. And we're moving out of the bloodstream and into the tissue in response to the chemicals being released in that injury and inflammation area. This allows our white blood cells to move to the site of injury or infection, and our white blood cells can then move there, start eating things up that are foreign, start eating up the bacteria by recognizing PAMPs, by recognizing things as non-self, and eating up and creating our inflammatory response that way. Later on in the inflammatory cycle, we've had our neutrophils doing their work, eating up whatever they can that's foreign, generally speaking. Later on, we'll attract our monocytes out of our bloodstream. They'll differentiate into macrophages or dendritic cells. We'll also bring in lymphocytes once they've been activated, our B cells and T cells. Our B cells will start creating antibody, and our T cells can start mediating the response as well as destroying any infected cells. We'll get into the details of that in chapter 16. The other important response in our inflammatory cycle is going to be the fever response. So fever is an abnormally elevated body temperature. And I want to make this very clear. This can depend highly on your age. When we get to dealing with, geriatric patients, patients that are much older, their body temperature is naturally lower than yours. So for a geriatric patient, potentially, while we see 98.6 as a healthy temperature, for a geriatric patient, potentially their normal body temperature for 98.6 might be considered a fever in some cases in some geriatric patients. So this isn't a hard and fast rule. When you guys get into practicing medicine and you're dealing with your patients, you'll learn more about this, whether it is with animals or people, that this can be something that is not a hard and fast rule. Not all humans at all times is 98.6 degrees Fahrenheit a healthy temperature. No. So you'll learn about those scenarios when you get into practicing medicine more. But generally speaking, elevated temperature from what is considered your norm will be a fever response. And for most healthy adult individuals, this is going to be temperatures at about 100 degrees Fahrenheit will be considered fever. When you're sitting at home and you're getting that thermometer and it's saying 99, you're like, you're not feeling well and it's 99. If you go to the doctor, man, with a 99, they're going to look at you like you're crazy. They're going to think that, man, you don't have a fever. What are you talking about? So 100 is usually the mark for otherwise healthy individuals. For low grade fever, 100 to 101. Once you get into 102, we're talking moderate fever, and high fevers are marked at about 104. If you're ever having a high fever at 104, or if you have a fever that's 103 and it's lasting for more than 3 days or so, then you need to be going to a doctor to go get medical help. Because once you get up above 105, we start denaturing the proteins in our body. And the first ones to go in that is usually proteins that are in the brain. We already know that your nervous system, especially your central nervous system, it can't repair your nerve cells. Once they're gone, they're gone. You can have permanent brain damage result from a high fever. High fevers can be deadly, and it's because of the denaturation of those proteins, the proteins that have functions, they are meant to function at 98.6 degrees, and 105 start interfering with our hydrogen bonds and the way that the proteins interact with each other. And the chemistry of that can be devastating to how your body functions. So absolutely, if you've got a high fever, you should be going to see a doctor and getting that fever under control. Pyrogens are chemical signals that initiate fever response. These chemicals can come from within your own body. Your white cells might be seeing things around them and saying we should make a fever, and they'll make chemicals like interleukin and TNF that are going to signal to the body that we need to have a fever. Those are endogenous pyrogens. When they're made by your body and your cells, your human cells, to signal a fever, it's an endogenous pyrogen. If it's exogenous pyrogen, this is responding to special markers that are seen on our infectious agents, things that are associated with those infectious agents. Some of these can be like PAMPs and things like that, can act as exogenous pyrogens. This includes endotoxin, which we've talked about so many times, we're probably getting sick of hearing about it, but endotoxin, which is lipopolysaccharide or LPS essentially on the surface of our gram-negative cells, that can just instigate immediately a fever response. So fever, what's the purpose? I'm sure you guys have just been waiting with bated breath to hear about why we have fevers and why it's important. So essentially, fever is going to increase the temperature in your body, which can inhibit the growth of some microbes. So that raised temperature of your body can inhibit microbes, some of them, from being able to survive well, especially environmental opportunistic pathogens. So our environmental opportunistic pathogens, they don't do very well in the elevated temperatures of the body. They are meant, to exist out in room temperature. And so that elevated temperature is just going to hinder them from growing even further. If you're a true pathogen, however, this might not be enough to stop you. It'll also impede nutrition to bacteria. Bacteria also need iron in order for their proteins to work effectively. And essentially what will happen is in the fever response, we can hoard away the iron away from the bloodstream. So it's not available free to the bacteria. So that will slow down the enzymatic functions of the bacteria from hoarding away the iron. It'll slow them down because they can't grow because it's too high of a hot temperature. That's how that we can affect the microbes. But fever also benefits our metabolism. So raising our body's temperature raises our metabolism in general. It increases hematopoiesis, so making our white blood cells from our stem cells, increases that. It increases the phagocytosis activity. and it speeds up our adaptive response and helps our lymphocytes get to where they're going faster as well. So fever has all of these really great benefits. So should we treat fever? Well, depends, exactly. So this is something that depends very much so on your specific situation. And of course, best scenario would be to get guidance from your doctor. However, if you're just got a mild illness and you're feeling a little down, but you can manage the pain, and you've got a fever that's under 103, and then you might consider letting it go. However, once you start getting into that higher moderate range of a fever, or if your moderate fever lasts more than 3 days, or of course, if you're getting up to a high fever as an adult, you should be seeking medical care at that point. If you have a child with a fever, you should probably always be seeking medical intervention with your child because their systems are a little bit more delicate and might not be able to manage these fever responses as well. And so treating with medication like Tylenol and things like that to decrease the fever might be an option in that case. But next time that you're sick, you think about this. You don't, just because you have a fever doesn't mean you need to stamp it down. All right. So interferon is our molecule that is involved in signaling the neighboring cell that we have a viral infection going on. So interferon interferes with the viral infection. So if you're infected, you've got a virus, you've got an invader coming into your house, you call up your neighbor, you tell your neighbor, get your defense mechanisms in place, and then their neighbor will be prepared, hopefully a little bit better than you were when you got initially infected. And so this can definitely have a big impact on slowing down viral infections. And we can even use interferon to treat certain viral infections to help amp up the normal response in a viral infection. All right, moving on to the complement cascade. This is a huge mystery to a lot of people, but it doesn't have to be. Complement is a set of about 50 proteins in your blood that's just there all the time. And you think about this, it's sort of like bombs that are just hanging out in your blood at all times, but they're neutral. They're waiting to get activated. But they're fine, they're safe until they get activated. Once one gets activated, one bomb goes off, it can cause a cascade of all the other bombs nearby and setting off, like lighting one match. And then that leading to, if you have a whole handful of matches, lighting one of the matches leading to all the other ones getting lit. That's a cascade response. So the complement reaction is basically a cascade response. Once you activate one complement protein, will lead to a cascade of all the other proteins in your bloodstream. There's over 50 different proteins involved in the complement cascade. And it will eventually, once you've activated one, can lead to amplification throughout the system. There's 3 ways that you can activate the complement cascade. The first is called the classical pathway. If there is an antibody bound on something, that can activate the complement cascade. So classical pathway is activated by antibody. The second pathway is the lectin pathway. The lectin pathway is when we have a protein called a lectin that recognizes microbe sugars on their surface, things like mannose. We don't have these on our normal cells. So these lectin proteins, if they bind to a bacterial sugar on the surface of a bacteria, that can activate complement. So that's the lectin pathway. The third pathway is the alternative pathway. And this is usually repeating molecules on microbes. This would be things like LPS and potentially peptidoglycan, but these sort of similar to PAMPs. But remember, PAMPs are recognized by our white blood cells. In the alternative pathway of the complement pathway, these are just proteins reacting and changing shape when they see things. So here, very similar to PAMPs, but we're talking about individual molecules causing proteins to change their shape when they see these molecular structures. So the classical pathway is activated by antibody. The lectin pathway, when the lectin protein sees microbe sugars. And then the alternative pathway is other repeating molecules on microbe surfaces. Very common one, very obvious one, LPS or endotoxin can trigger this pathway. So once you've activated complement, what happens next? In the complement cascade, our main steps are going to be initiation. This is lighting the first match, essentially. An initiation, this is like lighting that first match or setting off the first bomb. So we have, through either our classical pathway our lectin pathway or alternative pathway. We've either seen antibody, we've seen a sugar on a microbe surface through lectin binding to it, or we've recognized like LPS. That's happened. We've lit our first match. That's the initiation step. Now that we've set off one bomb, now that we've lit one match, now the cascade and amplification process happens. This is going to lead to all these other various proteins in the 50 proteins in your complement pathway to respond, to change shape because this guy changed shape. We're going to break off a part of us because we changed shape. Now that part breaks off and goes and interacts with this guy, and that's going to cause all these other slew of events to happen. You've lit one bomb, it's going to cause all these other bombs to be set off. That's the amplification and cascade effect. One match leads to lighting of many others. All right, then we have the polymerization step. And the polymerization step, essentially all of these parts, these bombs that got set off, some of those parts will come together and bind with one another. And essentially what happens is all of these bomb parts, they're coming to bind together. These are the bomb parts. These are our complement proteins that got excited, that got activated by seeing lectin bound to a sugar, by seeing an antibody somewhere or something like that. And they're going to polymerize. After our amplification and cascade happened, we got excited, they polymerize, and they form a hole on the surface of a bacterial cell. And that's pretty easy to see how now these guys are essentially going to bleed out. And we've also created an opening where, now we can no longer maintain the homeostasis of the bacterial cell, right? That cell can't maintain its salt content, can't maintain its contents, so it can bleed out and things can leak in as well. This is the membrane attack complex, or the MAC. Yes, this whole thing is. So we'll polymerize those excited proteins from the cascade. They polymerize, they come together and make a big old unit that forms a hole in the surface of bacteria. So that's one way that the complement cascade can work. All right, so that's the first way, creating that membrane attack complex by polymerizing those things. Complement also has a couple of other functions. It can also help initiate inflammation. It gets involved in calling cells to come eat things up. So phagocytes, it can start calling them to be activated. It can start sending out signals to engage in the inflammatory response and help lead other microbes to where they saw the microbe. So they start sending out chemical signals, basically, and then other white blood cells can follow the trail, the scent of the chemicals to where they saw a microbe. So these are just proteins that are seeing antibody, sugars on the bacteria, or they are potentially seeing, LPS or some general molecules that indicate a bad guy. Once those proteins have seen that, they change their shape and can break off parts and cause other guys to change their shape, break off parts, and cause this cascade, this amplification of the reaction. And now white blood cells can follow that trail of that cascade to where they saw the microbe initially. And that can lead to the initiation of inflammation or amplification of the inflammation response. The third way that complement acts is through a process called opsonization. Opsonization is a fancy word for basically once we've activated some of these proteins, they can stick at the surface of this bacteria. And what did we learn in lab that a lot of bacteria have that help with them to evade our white blood cells? Do you remember what that structure was called? A capsule, exactly. So we have capsules on the surface of bacterial cells a lot of times that can evade. It makes the bacteria slippery and hard to grab onto. Once we started our complement cascade, these excited protein parts can stick onto the surface of those capsules and make it a lot easier for our phagocytes to grab on to those handles. Essentially, they act as handles for the white blood cells to attach to. And that's called opsonization. These are molecular handles that are made as a response to the complement, basically complement cascade. All right. Cool. cool. There is a video. I'm not going to play it now, but if you want to like put this in better visual perspective, I just don't have the time to play it